work. Sorry. Sorry, just some technical difficulties. So yeah, contaminants in northern ecosystems have been studied for a very long time, and our understanding of various pathways through which they end up in high latitudes have advanced over the last few decades quite a bit. In fact, multinational collaborations such as the Arctic Monitoring and Assessment Program established in 1991 have provided regular updates on the status of various contaminants in northern environments. And more research are also being conducted here in Canada through post-secondary institutions, government agencies, and of course, the Northern Contaminants Program have been crucial to our understanding and monitoring of emerging contaminants in Canada's north. Collectively, these monitoring programs have been very important to monitor, sorry, um, to monitor contaminant levels in animals such as seals, fish, polar bears, as they are critical for subsistence fisheries, hunting, and conservation, of course. So before I delve into the main focus of my talk, I wanted to quickly go over like how do contaminants end up in Arctic ecosystems, and specifically focusing on the long-range transport, or briefly talking about the long-range transport, but also regional and local environmental changes, which can be important sources of contaminants. In terms of long-range transport, a lot of research has been done to understand the specific mechanisms on how various contaminants move through the latitude and eventually end up in the Arctic, specifically the roles of various physical conditions such as temperature, circulation patterns have greatly contributed to our understanding of contaminants reaching northern ecosystems, where often they are not produced nor used. The second form that I'll be talking about, or I have here, is from local and regional environmental changes. And most of the examples I've provided are from Canadian North. However, it is not to say only Canadian North is impacted by these stressors. Other northern regions are also impacted by these stressors. So the regional sources can be related to local disturbances. For example, St. Pierre et al. Demonstrated that both mercury and methylmercury concentrations were higher at sites that were downstream of retrogressive thaw slumps in the Peel Plateau region of the Northwest Territories, while Peenpon et al. Used a paired lake study design to show that metals and polycyclic aromatic compounds were higher in lake sediments that were impacted by slump impacted lakes near Inuvik. Furthermore, in a recent study by Pelletier et al. Have shown that regional forest fires can also be key sources of contaminants to lake ecosystems. Or in some cases, contaminants could have been transported from southern latitudes and deposited into glaciers, and the melting of glaciers could be introducing contaminants into lake ecosystems, as shown by Sun et al. On Lake Hazen. So these are all important sources of contaminants, and our understanding on these region-specific processes are continuing to grow as a result of some amazing ongoing research programs. And again, these are just a few examples. There could be a lot more examples, including the development of urban centers, which could be contributing contaminants to northern environments, but also waste disposal, just to name a few things. In this presentation, however, I'm going to chat a little bit about how natural resource extraction operations can be significant sources of contaminants to local freshwater ecosystems, and how they could leave long-term biological damage, as my area of interest is biological responses to contaminants. Although I'll be talking about a Canadian example today, that is the gold mining operations around Yellowknife, this is not to say it is restricted to Canada's north. There are resource extraction operations occurring in other parts of the Arctic as well, and most famously the nickel mines in Norilsk in Russia. So mining in the Northwest Territories has a very, very long history, with more than 88 mines operating since the early 20th century. So Yellowknife is located on the northern shores of Great Slave Lake. It's indicated by a star there. And I would also like to acknowledge that the Yellowknife region has been the home of the Veleta Yellowknife stenai for centuries, and the research I'm going to be presenting to you today are from their traditional home. So gold mining in Yellowknife has a very, very long history. The gold bearing arsenopyrite ores were initially discovered in this region around the turn of the 20th century. However, detailed surveys and claims for various takes did not begin until the 1930s, with the first major mine, that was Con Mine, opening in 1938. However, due to World War II, the gold mining operations were briefly halted, and after the war ended, a new era in Arctic mining began when gold mining operations started at the Giant Mine. And this is because the sheer magnitude of production, but also the environmental legacy left behind by Giant Mine. So the gold around Yellowknife is extracted from arsenopyrite ores, which have to be roasted. Hence, during the first few years of operation, severe environmental contamination occurred as waste products, which is arsenic and arsenic trioxide, were released into the environment directly through the smokestacks. So as much as 7,000 kilograms per day of toxic arsenic waste was released through the smokestacks. However, during the 1950s, emission controls were implemented gradually, and that resulted in substantial reductions of arsenic and arsenic trioxide being released into the surrounding environment. Fast forwarding several decades into the new millennia, when the mines were placed under receivership of the federal government, remediation has now begun as well. So recently, post 2010, a lot of lakes around Yellowknife were sampled to assess contaminant concentrations, and particularly those related to arsenopyrite ore roasting. And in this study by Huben et al. Showed that contaminant concentrations decreased with increasing distance from the roaster stack, which suggests that uh, the gold mine or past gold mining operations have left an environmental legacy through elevated levels of contaminants in the water column. So increasing the number from 25 lakes studied in Huben et al. To more than or close to 100 lakes, Palmer et al. Showed that lakes around Yellowknife, specifically those around the mine, had the highest arsenic concentrations, and again, as distance increased, concentrations also decreased. This is a bit worrisome because for the protection of aquatic life, or the guideline for the protection of aquatic life and drinking water quality in Canada is 5 and 10 micrograms per liter. However, the concentration closer to the mine are several magnitudes higher in some of these lakes. So at this point, I would like to take a quick pause on mining contamination to discuss a couple of other co-occurring stressors that are impacting lakes around Yellowknife. So specifically, the development of the city of Yellowknife, because as the gold mining industry grew, this region also grew as a northern hub. In 1967, Yellowknife was declared the capital of the Northwest Territories, which resulted in substantial population growth. With urbanization and related land use changes such as sewage and waste disposal, electricity generation, construction of roads and houses, they can influence aquatic ecosystems. For example, the increased runoff from catchment can increase sedimentation rates and can also bring nutrients, to name a few potential impacts. And I hope I wouldn't have to convince anyone here that the Arctic is warming across all season. This data is from AMAP, and specifically in the cold season, the increases in temperatures are much higher. But again, all seasons are warming, and this is consistent with the trends that we are observing in Yellowknife as well. So the annual temperatures are increasing, but also the greatest amount of increase is occurring in the winter temperatures, while spring and summer are also showing notable increases. And a warmer climate has important implication to the functioning of lakes as well. So through changes in ice cover patterns, desiccation of some exceptionally shallow water bodies, or changes in thermal stratification patterns, and also warmer surface water temperatures are just a few ways through which the physical characteristics of lakes can be altered in a warming climate. So we have got a lot of co-occurring stressors. So the mining pollution, but also land use changes and climate warming. And how are these impacting aquatic biota? And that's the main focus of my own research. However, I would also like to acknowledge that biological responses to multiple stressors could vary through time, and we need to have baseline conditions to infer potentially complex biological responses. And unfortunately, long-term data incorporating pre-disturbance periods and continuous biological monitoring data for multiple sites are not often available for most regions. So we rely on natural archives like lake sediments to infer past environmental conditions and associated biological responses. So that's where paleolimnology comes in, and lake sediment archives uh, environmental information from both outside the lake, such as metals from mining uh, activities or other industrial activities, but also within lake substances or autochthonous sources, which are the biological organisms which leave identifiable remains in lake sediments. So these can be used to reconstruct past environmental change as they are continuously accumulating 24 hours a day, 7 days a week, 365 days a year, making them an excellent history book. So we usually go to lakes, try to retrieve a sediment core with a gravity core. We are very happy usually when we can retrieve it in the first go, but sometimes it takes multiple tries. But eventually, once we get it, we bring it back to the lab to assess various proxies, specifically looking at biological proxies such as algae that leave identifiable remains like diatoms, but also zooplankton and midges um, can be looked at as well. Furthermore, we also look at chemical proxies such as metals, as I have mentioned previously, but also isotopes. So the first study that I would like to share, which I was not directly involved with, however, the two panelists here today, Joshua Thienpont and Jennifer Carosi, led this study on Pocket Lake, which is within the Giant Mine lease property, and it's a perched lake, and it's only 1 kilometer from the roaster stack. So it essentially should track the mining or the gold mining pollution from the mine. So just a quick note about the data presentation here. So these are stratigraphic profiles presented with time on y-axis. So at the top, the more recent sediment, and as we go down, it's the older sediments and pre-disturbance periods and whatnot. So here we have got a stable, the metals profile for this region or for this lake, sorry, and it's relatively stable until mining operations begin, when things start changing. Particularly, the increase in arsenic is quite staggering, about 1,700% increase in arsenic concentrations. However, in the more recent sediments, they have decreased, and that trend is seen for multiple elements in this sediment profile. So now looking at the biological responses to severe metalloid contamination, again, Thienpont et al. Looked at 3 different trophic levels. So starting with Cladocera, which are keystone zooplanktivorous herbivores, and the metalloid-tolerant Daphnia taxa increase in abundance initially. However, with extreme contamination, the Cladocera as a whole group are functionally lost in the lake sediments. Similarly, for diatoms, planktonic diatoms, which are the Discostella Staligera complex, decrease while metalloid-tolerant Navicular taxa and Denticular taxa increase with metalloid contamination, and chironomids, which are often tolerant of various pollutants, have also undergone notable changes with increases in metalloid tolerant taxa. So clearly, the Pocket Lake chemical and sedimentary chemical and biological proxies are tracking the impacts of past gold mining operations. Interestingly, however, we do observe an increase in via visible reflectance spectroscopy inferred chlorophyll. This is an approach, a very effective approach to track whole lake primary production trends. And it's increasing through time, which may be a response to lengthening of open water growing season, which I'll be talking about a little bit further down in the presentation as well. Now, using paleolimnology to track regional trends across several lakes, and that's what Cynthia Cheney and colleagues did um, by looking at 20 lakes, varying distance, about 40-kilometer radius, and looking at the sediments for metalloid contaminants. And specifically, now, if you remember the stratigraphic profile I showed for Pocket Lake, there are a lot of elements and a lot of changes happening. So it could be quite challenging to show 20 profiles here. And Cheney and colleagues did an amazing job at elegantly summarizing this large data set into a simple plot as this one, where they have grouped all the pre-mining sediments and looking at the concentration of various contaminants. So in this case, arsenic, lead, and antimony, as those are the main contaminants of concern in this region. And specifically, looking at the pre-mining period, they are relatively low. However, during the mining era, they increase. Again, arsenic, lead, and antimony, all of them increase. And then in the more recent sediments, they have decreased. However, they have not gone back to background levels. They are still elevated in the sediments, providing some indication that contaminant levels are decreasing, but it might take more time for them to go back to pre-mining conditions, potentially. Now, looking at biological data, how have biota responded to all of the, well, metalloid pollution, but also other stressors that I mentioned initially during my introduction for the region? And that's where I did some of my work looking at diatoms. They are excellent indicators of past changes in metalloid pollution, metalloid pollution as we saw with Pocket Lake, but also eutrophication and climate-mediated changes to lake thermal properties. And again, biological data, as we saw in Pocket Lake, can be very dense. So I have summarized the data using statistical approaches to make sure it is not overcrowded when we are presenting or when I'm presenting this information. And essentially, the goal was to answer two questions. Have biota changed? And after mining activities ended, do they signs of any recovery in terms of going back to pre-mining assemblages? So I used a direct ordination approach called DCCA, or Detrended Canonical Correspondence Analysis, which has been used quite extensively to summarize data or multi-species biological data. And using this approach, we can estimate species compositional turnover as beta diversity scaled in standard deviation units. And then I used cluster analysis to determine the timing of change, again, looking at the full datum data. So before I delve into all the other 9 of the lakes, I thought it would be nice to start with the Pocket Lake, which showed the most amount of change, not surprisingly, as it was so severely contaminated by arsenic. Again, the changes around the mid-20th century, around the same time when mining operations peaked, is when the most amount of biological changes were observed as well. Now, moving on to 2 other lakes, Niven and Cam Lakes, they are urban lakes within the city of Yellowknife. They also recorded notable changes, and we can see substantial amount or the timing of changes occurring around the mid-20th century, but also more recently around the turn into the new millennia. Looking at a few more lakes, again, the timing of change is occurring around the mid-20th century, and they're all changing quite a bit. The further away we get, so for example, sites that are further up north, about 20 kilometers or so from the mine, the amount of biological changes that we are observing are more modest. They are still happening based on the specific stratigraphic profiles, which can be found in a series of papers that we have published from this region. But overall, the magnitude of biological changes varied across the region, and the lakes that were closer to the city and the mines recorded the highest amount of beta diversity changes, to summarize it. And I thought it would be a nice way for me to like finish up this presentation with one specific case study from this region, looking at the impacts of multiple environmental stressors. So algal blooms have been observed at Jackfish Lake. It is a lake that is located within the city, and post 2013, algal blooms have been a recurring occurrence in this lake. So the question was, have similar algal blooms occurred in the past, and what limnological changes that might be happening in this lake could make these recent algal blooms to occur at this site? So as I said before, it is located within the city limits. On the northern shores of this lake is the Northwest Territories Power Corporation Jackfish Lake power or diesel power generating plant, and it uses the water from this lake to cool down the generators. But a bit further up or northern to this lake is the Yellowknife solid waste disposal site. So again, we used the VRS chlorophyll method to track whole lake primary production trends, and in fact, substantially higher amounts of chlorophyll was recorded in the most recent sediments that is the period with known algal blooms. And the recent level of primary production is unprecedented within the context of at least the last 200 years, probably longer. So now what limnological changes are occurring, making this lake susceptible to these algal blooms? So that's where we looked at multiple different proxies, because again, the benefit of using multi-proxy approaches in paleolimnology is that we can gain insights on different limnological stressors. So first, to begin with, cladocerans, Daphnia taxa, which, as I mentioned earlier, are tolerant of metalloid pollution, increase, while bosmina, which are not tolerant of specifically arsenic pollution, decreases in abundance. However, when we look at the diatoms, we observe an increase in eutrophic taxa uh, that are indicative of nutrient enrichment of this site in response to local land use changes. And finally, when we are looking at chironomids, there is an overall decrease in profundal taxa that are usually indicative of well-oxygenated hyperlimnetic conditions, and an overall increase in littoral taxa. So what might be going on there? So eutrophication from land use changes around this lake could have contributed to the increase in primary production. However, climate-mediated changes to lake thermal properties, for example, longer uh, ice-free period, which could lead to longer growing season, can also favor higher production. And the decomposition of algal biomass, the bottom of the lake, can lead to low oxygen conditions to form, which could potentially explain why we are seeing an overall decline in profundal chironomid taxa that are often indicative of well-oxygenated hyperlimnetic habitat. So what could this mean for the recent algal blooms? So eutrophication from past land use changes, as I said earlier, but also warmer temperatures and also longer growing season. Uh, Something I have not shown here is the wind speeds have also been declining, so which all together creating the favorable conditions for buoyancy regulating organisms such as cyanobacteria. So overall, the contaminant concentrations are still high in lakes around Yellowknife, and the freshwater biota in Yellowknife were impacted by past mining activities and urbanization. And the changing climatic conditions will likely influence future biological trajectories in this region. Now, taking a step back, looking at Northwest Territories, there have been 88 mines that have operated, or at least 88 mines that have operated since the 20th century. And while the environmental legacies may not be as substantial as what we observed in Yellowknife, there's a need to understand how these legacy mines or abandoned mines in some cases can have an impact on local aquatic ecosystems. And then taking even a step back further, um, resource extraction is not just unique to northern parts of Canada, it's also happening in southern latitudes. And understanding how biota have responded to these contaminants is critical to protect and conserve aquatic biodiversity in Canada. Finally, for future directions, I think there are a lot of exciting research happening, and a lot of these research work is informing us how or what we could potentially expect with a changing climatic condition, and specifically looking at things like metal mobility in sediments and the interactive effects of diverse contaminants and the impacts of other legacy pollutants are the forefront of research, but also changing hydrological conditions and ice cover regimes and how they can impact uh, metal uh, mobility in lake sediments is also another growing field of research, as it can have important implications to the recovery of these aquatic ecosystems. And finally, there are more emerging pollutants like microplastics and how they are ending up in northern ecosystems with very limited information to um, uh, build policies for future. With that, I would like to thank a lot of people. This was a team effort and collaborative work from a lot of people from various institutions. And I'll be happy to take any questions or start the panel discussion soon. Thank you.